So this is the second of three linked pathways, glycolysis, citric acid cycle, electron transport chain, in that metabolism of glucose to pyruvate. Pyruvate is then converted to acetyl-CoA, and then it can enter the citric acid cycle. This is a cyclic cycle, and I didn't click on my pen here, with that results in the regeneration of what we're going to call a sparking compound. And that sparking compound is a 4-carbon substrate, and it is recycled in the 8 steps that encompass the citric acid cycle. You might have heard the citric acid cycle also called the tricarboxylic acid cycle or the Krebs cycle. Hans Krebs is responsible for elucidating this pathway, thus the name Krebs cycle. But it is a second pathway in this process that is devoted to generating ATP in some form, or ATP-like molecules, and reduced NAD or NAD plus and FAD molecules, which are then find their way to the electron transport chain and give up their protons and electrons. So, as I just mentioned, the citric acid cycle also produces high energy compounds for ATP synthesis, and that is that NAD reduced NAD and FAD, and again some ATP-like molecules. This is a schematic of the citric acid cycle. It begins with a 4-carbon compound that's called oxaloacetate. It combines with the acetyl group from acetyl-CoA to form isocitrate, not iso, citrate. It is just citrate here. So 4 carbons plus 2 carbons condensed together, and we get this 6-carbon citrate. So the highlights of this cycle are the reduction of NAD plus, and we have 3 of those. We've got the reduction of FAD to FADH2, and we produce 1 GTP. So this is guanine triphosphate. Also note, 2 molecules of CO2 are released, and those are exhaled through the respiratory pathway. So I basically said this previously, but the first step is going to be the condensation of the acetyl group from CoA reacting with the 4-carbon substrate, which is oxaloacetate. But these are the highlights of the cycle. We produce 2 CO2 molecules between steps 3 and 4, so we condense those into a 6-carbon molecule, and then we start taking, we take 2 carbons away in subsequent steps. We produce 4 reduced coenzymes. And the steps are listed here. You don't need to know those details, but you need to know the overall products of this process. And 1 mole of GTP, which again is an ATP-like energy molecule. So this is the same cycle, but now it's named, we've given names to the sparking compound, oxaloacetate. Here we have the acetyl-CoA condensing the 2-carbon with the 4-carbon to make citrate. And we'll go through these stepwise. There are, of course, enzymes involved in these reactions. You don't need to know the details of the process, but I would like you to see how intricate and complicated it is. It's quite amazing the what our body and what goes on in the cells of our bodies. So here's that first reaction, oxaloacetate. We have to hydrolyze that thioester bond. So we have water here, and we release coenzyme A and form citrate. Citrate synthase, so synthesis of citrate, that's the enzyme involved here. In step 2, we're just isomerizing or rearranging the location of that hydroxyl group. Okay, so here in step 2, citrate is a tertiary alcohol. Okay, so this carbon is bonded to 3 other carbons and the hydroxyl group. So this is rearranged and this hydroxyl group moves down to this carbon, which is bonded to only 2 carbons. So we now have a secondary alcohol, and this is isocitrate. And this reaction is catalyzed by conatase. The first decarboxylation occurs in step 3, and this is the process of converting isocitrate to alpha ketoglutarate, which has 1 less carbon than isocitrate. So now we have a 5-carbon intermediate, 1, 2, 3, 4, 5, because we lost that molecule of CO2, and at the same time we reduced NAD+. So that's our first NADH that's produced. This enzyme is isocitrate dehydrogenase. So 2 hydrogens are lost to reduce that NAD+. So the secondary alcohol of isocitrate is oxidized by NAD+ to form a ketone. So now we have a ketone, or our carboxyl group, carbonyl group here in our 5-carbon molecule. Step 4, we release another molecule of CO2. So now we have gone from a 5-carbon intermediate to a 4-carbon intermediate, 1, 2, 3, 4. And notice that acetyl-CoA is also involved in this step, and it is linked to this molecule, and this is called succinyl-CoA. So release another CO2, and we oxidize alpha-ketoglutarate and reduce NAD+ in the presence of coenzyme A to form succinyl-CoA and NADH. Alpha-ketoglutarate dehydrogenase is the enzyme utilized here. And remember, the names of these enzymes tell us something about what's happening. The 5th step is the hydrolysis of that thioester bond of succinyl-CoA. And the result of that is the intermediate succinate. And this process releases energy by converting GDP to GTP. Okay. So an energy producing step. Step 6, succinate is converted to fumarate, and this is done with the help of succinate dehydrogenase, and we reduce FAD to FADH2. in this process. This involves the addition or addition of a double bond between these two carbons. Two hydrogens are lost here to reduce FAD. And succinate dehydrogenase, again, is that enzyme. Basically, the second half of this cyclic pathway is rearranging the structure of these intermediates in order to reform oxaloacetate. In step 7, we are hydrating fumarate to form malate. So water is added across that double bond, and we create, or the product is malate, which is a secondary alcohol. So this is a hydration reaction. And the final step is recovering or reforming our sparking compound, oxaloacetate. So without oxaloacetate, the citric acid cycle cannot run. So two things are required, oxaloacetate and acetyl-CoA. So that secondary alcohol of malate is oxidized by NAD+, which of course itself is reduced to form the ketone portion of oxaloacetate. And again, the product is the regeneration of that sparking compound. So this cycle can start all over again. So this is a summary of the products, overall products of the citric acid cycle. 2 molecules of carbon dioxide, which again are exhaled through the respiratory system. 3 NADHs and 1 FADH2, 1 GTP molecule. So the main function of the citric acid cycle is to produce these reduced coenzymes. So they've been reduced, they've picked up hydrogens and they've picked up electrons, and they are going to transport those to the electron transport chain. And ultimately produce ATP. And that's what we're going to look at next, the electron transport chain.